ASG stands for antigen. Remember, these are molecules on the surface of the cell, a viral particle we have on our own cells, but those on other cells and particles are different than our own. And this is what your immune system targets for destruction. So the antigen, what makes a molecule antigenic is its size. Antigens are all very large molecules. They can be proteins, they can be polysaccharides, complex carbohydrates, they can be glycoproteins where you have a carbohydrate bonded to a protein, lipoproteins where you have lipids bonded to proteins, nucleoproteins, whatever. But they're very large molecules. And actually on the antigen, you have what are called antigenic determinants. This is the red. And these little shapes here, okay, represent the antigenic determinants. This is what antibodies reaction to, the antigenic determinants. And you can produce a myriad of different antibodies, each specific for a particular antigenic determinant. So one antigen actually can stimulate maybe production more than one antibody. It's got maybe more than one antigenic determinant on it. It's actually what the antibodies are responding to. And these blue things here, kind of Y-shaped, represent antibody molecules, which are often described as being kind of Y-shaped. And they bind here, physically bind or attach to the antigenic determinants by what are called antigen binding sites. So again, it's the antigen, or epitopes are also called, that stimulate specific antibody production. Again, it's very specific. As I said, many antigens have different determinants. So one antigen may result in production of more than one type of antibody. Now, the problem is this. There are molecules that are too small to be antigenic in their own right, but when they attach to a carrier molecule in the body, often a protein, they become antigenic. So many of our allergies to drugs, to food, to an insect sting, or whatever, the molecules themselves aren't antigenic, but when they attach this body molecule, this carrier molecule, they become antigenic. Those molecules that are too small to be antigen in their own right are called haptens. And this is why a lot of us have allergies, but different allergies. You may be allergic to a certain drug, like penicillin. You may be allergic to a certain food, like peanuts, or eggs, or shellfish, or something like this. But somebody else isn't. Because what you're doing is that hapten, that small molecule, is attaching to a carrier molecule, and now it becomes antigenic. And now you're stimulated to produce an immune response against it. And that accounts for the different allergies that people have. Whether or not that small molecule, that hapten, attaches to a carrier molecule or not. If it doesn't, you're free and clear. If it does, then you have the allergy. Same thing with poison ivy. Okay? Not everybody's allergic to poison ivy. Some people roll in stuff, no problem. Other people, they look at the picture and they're breaking out. I'm kidding. But they're very sensitized, okay? Because the toxin poison ivy is a hapten. In its own right, it can't cause the reaction, because we know it's poison ivy. But if it binds to a carrier molecule, then you become sensitized and you're allergic to poison ivy. You will break out.
Could you give an example of like a carrier molecule? Is it a carrier molecule?
It's a carrier molecule, usually a protein in the body.
Okay. So it would just be a protein that would exist on
yeah, it's a normal
membrane.
protein, but it picks up the hapten, and now the whole thing gets big enough now to become an antigen. If it's own right, the hapten is too small to elicit an antigenic response, or an allergic response. Only when it couples with big molecules can it do so. As I said, poison ivy would be a good example of this. The toxin of poison ivy is a hapten, but in some people coupled with a body molecule, usually a protein, then you become sensitized. And same thing with a lot of drug allergies. Not everybody's allergic to the same drug, and those that are, it's because the molecule, like penicillin, for example, is coupled to a large molecule, and now you become sensitized to it, and you can't take the drug if you have a allergic reaction.
I have a question about that. Why is it that you can take penicillin for like 10 years and then all of a sudden you get it and
Because the sensitivity may change, and some people too, like as children, may be allergic just about everything. I had a brother like that, you know. Kept me on the service, but anyway. But, no, he was allergic to practically anything. Plus he had polyps too, so he was a real mess. But he outgrew them. So your body chemistry changes, and it can change also as far as sensitivity too. And same thing as you get older too. You know, you may have taken this drug long, long, long time, and then your chemistry has changed somewhat, and now you become sensitized to it, and you can't take it anymore.
well, I had a lot of problems with my teeth and they gave me penicillin all the time.
Yeah.
And I was about
And you
23.
overdone it too.
Yeah, and then I spent 7 days in the
That's-
hospital. My
right.
whole face swelled up.
'Cause
I
you were
looked like
allergic,
a
yeah, you had it.
like an alien.
Yeah, you
And
had-
then when I had my son, he was born allergic to it, so they won't give it to him
Okay.
because they don't know if he outgrew it or not.
Right. Yeah, your body chemistry can change. It does change with age. Okay? And sometimes you outgrow allergies, so to speak. Sometimes you may acquire some later on. So it'll change. Okay. All right, characteristics of antibodies. Antibodies are all proteins. And capital A, lowercase d stands for antibody. Another name is immunoglobulin. Capital I, lowercase g. I'm going to tell you right now, I'm going to expect you to know the different kinds of antibodies. There will be a question on the test. It'll probably be multiple choice where I list the characteristics of the antibody and ask you which one is it. Okay, so learn them. All right, I'm telling you right now. Okay, first off though, antibodies have what are called antigen binding sites, like I just showed you. That's what physically couples with the antigenic determinant or epitope of the antigen. They're very specific. When we speak of antibody, we talk about valence of antibody. This is the number of antigen binding sites that the antibody has. And most human antibodies are said to be bivalent, which means they have 2 antigen binding sites. Bivalent, they have 2 antigen binding sites. And that's a typical human antibody. That's sometimes called a monomer because sometimes these smaller antibodies will combine together to form larger antibodies. Remember a monomer, when you talked about molecules, monomers were the building blocks to make up polymers. AP1, you probably talked about that. Okay. Okay, this represents diagrammatically an antibody molecule. It's often described as being kind of Y-shaped or T-shaped because it is flexible here. The stem of the Y and the arms of the Y. At the ends of the arms of the Y are the antigen binding sites. And this is called the variable region because there are many different amino acid sequences. Because what you have here are 4 polypeptide chains. 2 heavy molecular weight chains are called heavy chains, and 2 smaller molecular weight chains, which are called light chains, which are identical. They're held together by bonds. Okay, these are called disulfide linkages. You don't know what the name, but it holds the molecule in a particular configuration. All right, here at the ends of the arms of the Y is called, or are the antigen binding sites. And as I said, this is a variable region. There are a myriad of different amino acid sequences. So at least theoretically, we can produce antibodies against any imaginable antigen we may encounter during our life. Millions of different ones. Okay? But we don't produce them until we have the antibody. I mean, we have the cells capable of producing the antibody, but they don't do it until they become sensitized. So you have all these potential cells, lymphocytes, B lymphocytes, that are there, and they don't do anything until they've been stimulated. And then when they are stimulated by a particular antigen they come in contact with, then they will produce that specific antibody that reacts to that specific antigen. Okay? And where they bind is at the ends of the arms of the Y, the so-called variable region. Okay? The other end, the stem, is called the constant region because it has only 5 different amino acid sequences. And this determines the 5 different classes of human antibodies, which we're going to go into. This one I want you to know. Okay, so again, the antibody molecule, 4 polypeptide chains, 2 heavy chains, higher molecular weight, identical to one another, and then 2 light chains, also identical to one another, lower molecular weight. Okay? And again, where the antibody will bind the antigen is at the ends of the arms of the Y, antigen binding sites. And then at the other end here, the so-called stem region, that's the constant region, that determines the class of antibody. And if this attaches something, it will attach to a cell, but to a host cell. Okay? This can attach to cells that are parasitized and have foreign antigens on them. This will attach to normal cells. A lymphocyte, a basophil, a mast cell, and a eosinophil. We'll talk about later. Okay, so we have the bi-beta antibody. It's a typical human antibody. Has two antigen binding sites, but some antibodies consist of more than one bi-beta antibody, and those would be called multi-beta antibodies. Multi-beta antibodies. Okay, this just goes to what I've been talking about. The clearable region and the constant region. All right, the 5 different classes of human antibodies. And I'd like you to know these. You will see these on the exam. I'll say that right now. And they are designated by capital letters. And the prefix is Ig, which stands for immunoglobulin. Okay? So the first one is IgG. Immunoglobulin G. This is a monomer, which means it's a bivalent antibody with 2 antigen binding sites. It is the most prevalent antibody in normal human serum. Don't memorize the number. It may be 80% or more. But don't memorize that. It's just the most prevalent. So as for relative abundance, number 1 in the human serum is IgG. This one readily crosses blood vessels, gets into the tissue fluids to help fight infection. It can readily cross the placenta to go from maternal to fetal circulation. This is what provides passive immunity to the baby, because the baby gets those antibodies from the mother. It also protects against parasites outside cells, circulating viruses, bacteria. It also can neutralize the toxins produced by bacteria. It also is involved in complement reactions, stimulates and activates complement, including the lysis of cells, and enhances phagocytosis. So these are characteristics which I'd like you to know. So go through this, learn these, because you will see these, I promise. Okay? Next antibody is the biggest antibody. This is called IgM, immunoglobulin M. Okay? It's a pentamer. Pent means 5. So it's 5 of these monomers combined. Each monomer has 2 antigen binding sites. You got 5 of these. So how many antigen binding sites? 10. Okay? And notice, it's only the 3rd most common antibody in human plasma. Again, don't memorize the percentage. And notice too, it's the first antibody to appear with initial contact with the antigen. So when you first have an infection, you have a big rise or concentration or titer of IgM, and then eventually it's replaced by IgG. When you have a subsequent reaction, it's much more efficient and faster, it's mostly IgG. Okay? This is very effective in agglutination reactions like you see with mistransfused blood involving antigen binding sites. It's also involved in complement reactions like IgG causing lysis. It also enhances phagocytosis like IgG. And a few of these are found in B lymphocytes, but mostly it's in the blood. Okay. That IgA, the 3rd type of antibody, this one is a monomer when it's found in plasma. It's the 2nd most abundant in the plasma, but typically we find it in other body fluids, and here it's 2 monomers hooked together. It's a dimer, and now you've got 4 antigen binding sites. And we find it, for example, in our mucus secretions, our tears, our saliva. We also find it in seminal fluid produced by males, and in the breast milk, especially high in the colostrum, the first secretion from the lactating breast. Very important because it helps to protect the newborn. If that mother is breastfeeding, this is the antibody the baby's getting. It protects the newborn against what are called enteric pathogens. Babies usually get sick from diseases affecting the GI tract because of things they pick up. It's very effective to help you prevent the infection of these enteric pathogens, as they're called.
The IgM was the 3rd most abundant?
IgM is 3rd most abundant. Okay? IgA is 2nd. Okay, IgG is the 4th one. This also is a monomer, 2 antigen binding sites. only a small percentage of these in the plasma. It's primarily found attached to lymphocytes. You have your lymphocyte and your B lymphocyte. The antigen is attached by the stem to the cell membrane. The antigen binding sites are exposed. And basically it's an antigen receptor on our B lymphocytes. That's mostly where you find it. Okay, it's not involved in common reactions, does not cross the placenta. And lastly, IgE, immunoglobulin E. This also is a monomer to antigen binding sites. It's the least abundant in the plasma, as you can see. It's also found primarily attached to host cells, but not B lymphocytes. Instead, to mast cells, basophils, and eosinophils. And again, it attaches the same way. The stem of the molecule attached to the host cell, antigen binding sites are exposed. When they come in contact with those antigens, that causes these cells to release inflammatory chemicals from the cytoplasm. That's what brings them on the signs and symptoms of what we know as allergies. Okay? Okay, we're out of time. We'll stop here. Thank you for sticking